The first line of defense against anything foreign, including evading stealth pathogens. This defense system enacts the complement cascade, as it's known, to promote identification and removal of foreign substances. Inflammation is one of the first responses of the immune system to infection, which is not necessarily a bad thing. The issue becomes when that inflammatory signal is left on for too long, thus leading to tissue damage. Pattern recognition receptors, such as toll-like receptors, C-type lectin receptors, NOD-like receptors, and RIG1-like receptors, assist in distinguishing self from non-self and identifying pathogen-associated molecular patterns, which then release these inflammatory mediators, such as tumor necrosis factor, interleukin-6, and interleukin-1. This process tags pathogens for amelioration. Naturally, many pathogens have evolved mechanisms for immune evasion, in part by mimicking host cells so the immune system does not recognize them as foreign, which brings us back to the importance of glycoimmunology. Sitting at the upper echelon within the innate inflammatory response systems are CEACAM1 and the Annexin genes. CEACAM1 controls the initiation of the modulation of innate and adaptive immune responses, while Annexin controls the shutoff switch when the inflammation has served its purpose. And of course, involved with all of this is a gene called SLC2A3, and it's important because it transports mannose, fucose, and vitamin C, which assists in the regulation of CEACAM1 and the innate immune system itself. We as humans are exposed to millions of potential pathogens daily through contact, ingestion, and inhalation. Our ability to avoid infection depends in part on the adaptive immune system, which remembers previous encounters with specific pathogens and destroys them when they attack again. Adaptive immune responses, however, are slow to develop on first exposure to a new pathogen. It can therefore take a week or so before the responses are even effective. By contrast, during the first critical hours and days of exposure to a new pathogen, we rely on our innate immune system to protect us from the infection. Through the study of glycoimmunology, having the ability to identify these stealth pathogens upon the initial infection is paramount. This is why when we combine things like mannose, bladderwrack, N-acetylglucosamine, and things that are high in vitamin C like sea buckthorn, then become powerful tools that work in synergy to support the innate immune response.